So now we move on to the next presentation, which is on pulse nutritional phenomics to combat global malnutrition and obesity. It is by Dr. Dil Thavarajah. Dr. Dil Thavarajah is an associate professor at Clemson University. Where is Dr. Dil Thavarajah? I'm not able to see. Can you just wave your hand, ma'am?
Yes.
Yeah.
Can
okay.
you hear me?
Yes, yeah, I'm just going to introduce you. I just wanted to see whether you are there or not. Yeah.
Right, I am there.
Dr. Dil is the phenomics component of the Feed the Future Innovation Lab for Crop Improvement at Cornell University. Dr. Thavarajah started the first USA Pulse Quality and Nutrition Laboratory at North Dakota State University, Fargo, North Dakota in 2010. Before these positions, Dr. Dil worked at the Canadian Lentil Biofortification Program at the University of Saskatchewan, Canada. Dr. Dil is internationally recognized as a leader in lentil biofortification, especially for iron and selenium. Dr. Dil advises graduate and undergraduate students, published numerous research publications, book chapters, and serves as an honorary visiting lecturer in University of Peradeniya, Sri Lanka. And she's a key partner to the ICAR's Lentil Biofortification Program. Ma'am, it's yours.
Thank you. Let me see. Dr. Varshney told me a lot of things to share. How do I screen?
Welcome, Dil. Thank you very much for joining. And this is very early morning for you, but thank you very much for joining.
Okay, perfect. All right. Thank you, Dr. Varshney, to inviting to do this because we are locked down. We only go to the field, and our labs are only open very small time just to do the essential work. Yes, it is little in the morning, but it's a beautiful morning, 6:30 in the morning AM. So the talk that I wanted to talk about how pulse nutrition, which is very important, but I listened to Dr. Varshney's talk in 2018 or 19 at Morocco IFLRC meeting. And one of the things he was telling us was genomic work is so cheap, but the bottleneck is what the work I do, the nutritional phenotyping, because it's a chemical phenotyping, it's very expensive, and we need to find a way how to go hand to hand with the genotyping work people like Dr. Varshney and Dr. Krasavich, all these people doing, but we have to feed the nutritional data into this pipeline. We had to find a way how to do that. So I wanted to talk about a little bit of work from chemical phenotyping, how we can move forward. I don't have much of the data right now because we just started moving from destructive phenotyping to non-destructive phenotyping. But next year, this time, I think most of the breeders around the world, ICAR AICRP-Pearl can use our techniques. So with that, first I like to acknowledge my team, Clemson Pulse Quality team. Without this, quite a bit of people, Dr. Shaipi is my in-house breeder, and Dr. Tava, Tristan, all these people, they help us to get it going under difficult situations, and our lab functioning very well. And our funding mostly for the federal funds from NIFA and USAID. We also developed the organic plant-based protein, which is the project funded by Good Food Institute and USDARS. And also we do like a community food and nutrition programs and helping gardening and having the proper legume crops with the leafy greens and the tubers, so which is also funded by NSF, National Science Foundation, and also Department of Agriculture and CAFLS. That's my college. And so these are all the people, and also ICAR does a great job helping us to keep moving with our lentil biofortification. Dr. Krasavich's team and our wheat breeders, Dr. Boyle, and everybody listed here, they all help to make this possible. If everybody can see, I just wanted to get this down so then everybody can see my, okay. So I wanted to start with something Dr. Norman Barlow said. food is the moral right of all who are born to this world. Dr. Norman Borlaug, 1914 to 2009. This is the father of the Green Revolution. And it's very interesting how he was putting together what is food security, a situation that exists when all people at all times have physical, social, and economic access to sufficient and safe and nutrition food, especially the situations like now, we all locked down. Are we all getting enough nutritious food? It's a problem. I know we will talk about that a little later. So in the food security, 3 categories that we wanted to talk about is the availability, access, and utilization. All these breeders around the world, they are doing such a great job producing food, but we need to make sure these are available to everybody, everybody has access, and they know how to cook and how to eat, what is the nutrient bioavailability. This is a complex situation. So, okay, here is the trends in micronutrient malnutrition. If I can move this bar so I can't clearly, okay. So this is the global overview of the malnutrition. Malnutrition is in all different forms. It could be micronutrient malnutrition or obesity or overweight. So you can see here from 2000 to 2019, the actually stunting is decreasing. But this is the overweighting. Overweight or the obesity is increasing and wasting is right here. So we have done, our plant breeders around the world has done quite a good job, and our epidemiology teams, nutrition teams has done quite a bit of good job to reducing stunting up to there. But then the new problem, the obesity is increasing. So we haven't done almost all the work. We still have a lot to do. So, okay, this is it's very difficult to get the slides out with this. Okay, so you can see here, this is the 7 sub-regions have the high or very high stunting prevalence. The purple ones, the red ones is the Southern Asia, and this the parts of Africa has the most severity. So, our goal should be Africa and Asia bear the greatest share of all forms of malnutrition. This is the micronutrient malnutrition as well as the obesity and the overweight. So we have still a lot of work to be done for Africa and for Asia. So when I look at the Sustainable Development Goal, this is the brand new report coming out from the UN. And there, one of the emphasis was like, how, when, and what? How can we move from 2016 to 2030? How we can sort it out, this stunting, also overweight, obesity, and wasting. So one of the first thing how we're going to sort it out is sustainable, resilient food system for healthy diet. So one of the thing is the healthy diet. So what can we do? Most of our biologists, agriculturists, food scientists, what can we do? So how about we use agriculture as the framework, and within that framework, we can have a diet rich with nutrient, not just nutrients. We need to consider the bioavailability frame. Also, agriculture, diet, how we can empower this. We need to empower the smallholder farmers, especially in Africa and Asia, because they contribute quite a bit, 95% of the agriculture production. And also we have to emphasize the nutrition. So it's just not only cash crops. How do we having range of crops, maybe cereals, legumes, tubers, bananas, and so that way we having a range of different types of food. We can have a diet that enrich with all the nutrient, and then empower the women, empower the mothers, and also then the outcome should be human health instead of the grain yield. Can we link the yield and the human health? So that is a kind of a so next, I like to share the components of the food system. This was actually done by Dr. Graham Welch in 2010 with Jerry Combs, which are my advisors that I really enjoy working with them and taking their legacy further. So in their model, I still use this model in my class as well, because haven't changed much since for last 2 decades. So all the component, public health, policy, biophysical, environment, economics, and everything is in there. And you can see production, acquisition, and utilization. This is where utilization, this is where the women and the mothers, they fit. So whatever the food come to the household, we need to make sure they prepare right and they feed their children. So that's why I really like the biofortification platform. The biofortification platform is introduced by the Harvest Plus 2 decades ago. So, and it is continuing, and my research program totally focused on lentil and field peas. We work on chickpea little bit with the Dr. George Vandermark from WSTU, but more focus for the lentils and the peas. So our platform, we make human health as the clear goal based on the sustainable development goal. Is it for all forms of malnutrition? It could be micronutrient malnutrition, or it could be obesity. Right now, we most focus on protein malnutrition and the carbohydrate, low digestible carbohydrate. And also not only that, and we have these breeders, they have these varieties, and we make sure these nutrition traits are in there, and what happened after cooking, cooling, or like how do you prepare at home, and then when you eat it, what happened inside the human body, and then we educate them. Keep continuing, keep doing. You can't stop. You have to continue to do this nutrition throughout your life to have a healthy lifestyle and not to have these non-communicable diseases. So we have, our motto is the improved health can be addressed by changing the paradigm for the agriculture. So what's happening now? Now is like the time that COVID-19 pandemic. So you see the actual model of the 2 decades ago, the Dr. Combs and Welch developed that model, but now the model is a little different. And because of the COVID impact and people producing, these are our organic pea trials in South Carolina, we are producing, but we need to make sure these things will be, and this is a smallholder farmers from rice farmers, and we need to make sure these things will be actually access to these people. And the problem, this availability of food, and if anybody is interested, this is a really good paper from Turner et al, and you can read it, and the model is development based on their his paper by the UN. And now is a problem with the accessibility, and also the production system is going to be disabled because of the lockdown. And we will facing threat increased level of malnutrition because all this food is difficult to go there, especially fresh food. So it is a good time if you stock up your pulses. This is a good time, rice, pulses, because most of the other fresh vegetables going to be a problem. So that is the pandemic, what we are happening. But I also like to show this. This is the figure that I got from Dr. Welch 2010 paper. There's not much change for 2 decades. And you can see this is the cereal production, okay? And here is the population growth, and look at the pulses growth. So there's quite a bit of gap in the production. I know our breeders are working very hard to increase the yield and match with the cereal yield. But cereal is the #1 thing, provide the energy for us. And to make it balance, you have to mix that with the pulses, basically to balance the amino acid profiles and provide quite a bit of other micronutrient into the diet. So there is a gap here. So that is for until 1999. So this is our data from 1961 to 2009. You can see how people eat. Actually, people eat a lot of fat. supply, grams per capita per day. And this is the sugar supply, kilograms per capita per day. And look at the pulse supply. So this is up to 2009. But if we go further beyond, this trend has not changed yet. I haven't looked at the very recent data. This was done a couple of years ago, like 2-3 years ago, but still the trend is not changing. So if you want to be a more healthy, nutritious diet, we might have to change this trend a little bit. And why? Because legumes were forgotten. And this Nature paper was absolutely right timing. And it was talking about neglecting legumes has compromised human health and sustainable food production. This is one of Dr. Varshney's team and Dr. Forrest's team. So like the really good number of authors that actually care for these legumes, and it shows us legume production is declining. So we have to do something about it, because that means we are losing a lot of nutritious food. So especially for Asia and Southeast Asia as well as Africa, we need to promote these crops. I grew up in Sri Lanka. I ate 3 meals of lentils. That's our main thing. It's just still growing up to now, and families, like, we need to promote this healthy food, because most people don't know around the world, except Asia and the, I want to go to the previous slide. Okay, I can go to the, so, and I really like this model. So how do we, this is the model developed by Dr. Varshney's team, I'm sure. So they're talking about all the gene discoveries and the breeding efforts. So in here, the phenotyping, we somehow need to incorporate our nutritional traits in there. So he put down here the infrared and the NIR technology. Yes, these are very good, because the breeders can select their crops at the field without like a 50 cents per sample. Chemical phenotyping is very expensive. So how do we put this into there, like the other traits? Because this is limited for some of the things, because you can measure protein, but you cannot measure protein quality. You can measure starch, but you cannot measure some of the specific quantities of the starch, because those are the things more important to make the healthy gut and things like that. But this diagram is very important. I would prefer like every breeder has that, and then incorporated some nutrition into their pipeline. So this is the work that, this is how our protocol works in my lab. So the South Carolina, we don't have pulses, and it is the brand new specialty crop. We start growing field peas from last year, that's Pisum sativum. So the 4 things, but I work with the breeders around the world. So 4 things that I work with the breeders is, yes, agronomic adaptability is very important. If you don't make money, the farmers don't make money, then it's going to be a problem. So yield, disease resistant, drought tolerance, nitrogen fixation. So those are like very, very important things, and quite a bit of genomic facilities and gene identification. It's almost all done for this. People are working on it. And then what about the major nutrients, that how we incorporated that into? So basically we are looking at carbohydrate, proteins are the #1, our major goal. And then we are moving into vitamins and minerals. And also don't forget about color, shape, milling and cooking qualities. Those are very important things into your breeding pipeline. So when you have your breeding pipeline, I would prefer you talk to the consumers and the farmers, everybody, and put that information into the pipeline. And then when you develop your varieties, how well they cook, because we need to make something that mothers like and the children like to eat. So all these things had to be in your breeding pipeline. So you cannot forget the nutrition education. So it is nice, each breeder talk to like a clinician, a nutritional clinician. So our program, we have a clinician we talk to, we have breeders, we have genomic experts. So it's like a collaborative effort with my lab that we work. It's not a single effect. So, so this is the pipeline that I was talking about, like we were working on my lab. So how we improve the nutrition crops. So most of the reference genomes are, So Dr. Krasovich team, he's the director for APT for the Clemson University. So his team helped us with quite a bit of the genomic work. And then my team, we have all sort of nutritional phenotyping system in the lab. We have from analytical chemistry equipments to the non-destructive tools, like this is the FTIR unit. Now we are moving from NIR to FTIR, which is more powerful to look at some of the protein quality work. And then we develop this kind of handheld one. This is actually developed by the Cornell University, Dr. Mike Go. And then this Pheno app developed by Jesse Poland. So we are working as a team. So how these chemical phenotyping at the end come into here, so the breeders can select their, I don't know, F4s or F5 single row plants in the field and discard them because, oh, this one has a better protein quality than this one. So if we can improve that, all the systems and the crop improvement going to be really well. But the problem is, like, this is the end. This is how people eat lentils, cook lentils. But what they like, the taste, quality, smell, all these things go back here, and this is a circle that never spends so long. And all the nutrition quality traits that we do, we have to incorporate that into the genomic and bio computational analysis. So this way we can identify the genes and their functions, and we can develop the varieties with all these qualities. So I want to do more emphasis on the food matrix. So this is the lentil seed. So when you look at the lentil seed, 60% is the carbohydrate, 25% protein, 2% fat, 3% ash, and this is the all 3% is the other nutrients that you all talking about, like micronutrients, vitamins, and everything in this 3%. So it is very important because pulses has this 6% ashes is the mineral, and then 3% is other nutrients. So this is a very important part. But also carbohydrate, which is we neglected and we always blame for, but it is a really good component too. Especially for pulses, they are called low digestible carbohydrate, and they digest in the system very slowly. So if you look at this, this is the how much nutrients you need for your body. So this is about 55 nutrients, like 9 amino acids, lipids, and minerals, and trace elements, and vitamins. So all those things in this little 3 to 6% right here, but quite a bit in here and quite a bit in there. So in your breeding program, you need to make sure that these people who are going to eat that actually have some of these nutrients. So some varieties like Harvest Plus objectives to increase the high iron, high zinc, and beta carotene. That is really, really good objective. Also now, then the next thing we need to look at the carbohydrate because of the obesity and the protein. So what is the quality diet? So quality diet, according to the WHO definition, is a healthy diet highlights the importance of starting healthy eating habits, so mothers, babies, and the families in early life. So if you learn how to eat pulses very early time, like I'm a good example, and then my child, like I teach her how to eat that. So therefore, then when you grow up also, that's how it grow. And then limited intake of free sugars or the added sugars and the salt. And if you look at, so this is a picture of our faculty coffee that I started introducing pulses to South Carolina, and they enjoyed quite well. This is when I first started in Clemson. Now, quite a bit of people eat lentils and peas, and mostly hummus is very famous in US. So if you look at all these crops, lentil, chickpea, field pea with rice, you can see quite a bit of protein from these pulse crops. And also this is iron. This is all the data that I generated from my lab and for the last couple of years. The most things right now we emphasize on this prebiotic carbohydrate, because prebiotic carbohydrate change the gut microbiome, and it helped to reduce the weight as well as the reduce some cancer and other metabolic disorders. and also resistant starch, which is the part of the prebiotic carbohydrate. And all of other things you can see, actually these pulses are quite interesting crops. So let's look at what is the prebiotic carbohydrate. I need to go previous. Okay, here is the prebiotic carbohydrate. So what is prebiotic? Prebiotic is defined by Dr. Gibson et al. In 2004, a selectively fermented ingredient that allows specific changes both in the composition and activity of the gut microflora. So that means these act like a substrate to the gut microflora. So if you eat the diet rich in prebiotic carbohydrate, that would help to have a better gut health. So this classification came in 2004, and then we used that into our lentil program. So in the carbohydrate can be divided into low digestible or digestible carbohydrates. And then the things that we work on, sugar alcohols in the orange, and then the resistant starch, RFOs. Some people cannot eat RFOs because of the flatulence and the gases, but RFO has quite a bit of health importance. A decade ago, most breeders like to knock down RFOs in their varieties, but now we know RFO has quite a big influence of the gut health, and also fructooligosaccharides. So these orange ones are the what we are interested on that, not only human health. One of my graduate students are right now working on it, because one of the graduate students who start with this, and he's a medical doctor now, and what we found was that was the human side. And later when we study, and we found these are important for the plant health as well. So one of the graduate students right now looking at the lentils and see how these carbohydrates actually help to plant to drought stress tolerant as well as the climate resilience. So some of these lines has high amount of sugar alcohols. They perform very well at the low moisture conditions. So still we are working on that. So can pulse-based diet reduce the obesity? So this paper came 2013 from the British Journal of Nutrition, from the Netherlands group who working on the gut health. What their idea was, their picture here, the overnutrition, or eating burgers, fries, and Coke, and all this overnutrition food, or like empty calorie food, what it will do, it will create the metabolic disorders and the inflammation in the body that will create the obesity. So this is their model. And also they said that having the gut microbiome, which is act on these key significant organs, it will help to reduce the inflammation and the inflammation and the obesity. So with that, we thought we have a hypothesis that can pulse-based diet reduce the obesity? So if a lentil rich in RFOs, fructooligosaccharides, resistant starch, can these utilized by the gut microbiome? And will that change the good microbiome, that is the bifidobacteria, and reduce the pathogenic bacteria? And if we can change the microbiome, it will change the metabolic composition, and ultimately it will reduce reduced obesity. See, one of the food science major student was in my lab, and we start pushing this hypothesis further. So this is the, if you look at this one, this is how the lentil prebiotic carbohydrate composition in lentils. So it's about it's about like 10 to 15 grams in 100 grams for the total prebiotic carbohydrate. And the recommended intake is about, if I can move this, sorry. So if I can move all the things that I can see the slides. So anyways, so what I'm talking about is the lentil has quite a bit of different prebiotic carbohydrate, and in 100 grams, it provides about 12 to 15 grams. That is the dry seed, but dry seed is not the one that people are going to eat. What happened when it's eaten? So what we found was these are the different lentil market classes. And we only look at for the resistant starch, and we look at the other carbohydrate as well. You can read these papers if you're interested, how these carbohydrate change when you're cooking, soaking, and cooling. So this is about the resistant starch, because one of the prebiotic carbohydrate helped to increase the bifidobacteria that was written in the literature. So we looked at the resistant starch. What we found was this is the cooking, and the blue lines are cooling and the reheating. You can see that the resistant starch amount almost died, 3 grams to 6.5. That is a big thing. So what we are talking about, like you cook the lentils today, and then you put in the refrigerator, you eat next day, you microwave it and eat next day, that increased the resistant starch almost double. So these cooking procedures and how you use that in the house actually benefit the nutrition changes. So I know it is hard to incorporate these into the breeding pipeline, but these are the things that breeders need to think about, because it's not the dry seed what happened after cooking and cooling. So at least if we have minimum number of carbohydrates or the resistant starch in the dry seed, we can predict it, how much going to be after cooling and cooking. So it's the same kind of trend we see for the other pulses like chickpeas and beans as well. So with that information, we thought, and how we kind of test that, we decided to go with the small rat study, because quickly way to find out whether these nutrients components are effective, we decided to go in the rat study. And you can read this paper, it's in the Journal of Agriculture Food Chemistry. We had three diets. One is the control diet, and the other one is the cornstarch diet, and the other one is the lentil diet. And then we had each group has 12 rats, and then we did this for 6 weeks in a control environment. And then we took blood samples, and then we took the feces samples, and we extract the RNA, and we look at what type of microbiome in there. So this is the diet composition. You can see actually, if you look at here, what we did was the control diet has 0 lentil, corn diet has 0 lentil, and 780 grams. That is actually 71% in the diet. So this diet was developed by one of the nutritionists, animal nutritionists from Clemson University. And then we feed this diet to 6 weeks. Just a quick study, because we wanted to know, is this prebiotic carbohydrates going to work? And here is the data. This is the 6 weeks time period and the 3 diets. You can see the red lines are the lentil diet, and it's after 6 weeks compared to the corn and the control diet, it was quite significantly different body weight. So that was promising. And so then how is the body composition? So this is the body fat. Look at the control diet versus lentil diet, which is significantly different. This is in 6 weeks. So actually lentil is really, really healthy. And this is the liver weight. We also got the liver weights, and we wanted to see whether fatty liver developed, and we want to take the tissue samples of the liver. So we did not see significant difference of the liver weights, but the lentil diet has some reduction. And this is quite interesting. This is blood plasma triglycerols. And you can see lentil diet has quite significant, similar to control, but the corn has significantly high. This data is from 6 weeks. What we need is a larger clinical trial to do that. But this, I wanted to make sure, is this the promising work? So that's why we did this study in the lab. And then here is the abundance of the microorganisms, what we see. And what we see was the good bacteria, that is the Firmicutes and Bacteroids, was about 46 to 73 and 25 to 24%. And then we saw the Clostridium were decreasing. and quite a range of the bacteria phylum. All the statistical analysis is in that paper. You can see that some of the bifidobacteria species were significantly increased in the lentil diet. But actually these were done in a clean environment. We took the feces samples and extracted the RNA. It would be important, like that's the only way we could have done, but there's quite a bit of things that we could have done, but it's a quick study. We want to know whether prebiotic carbohydrate works. Actually, it did work. So, and then we took a liver sample and look at the, this is SEM images and looking at how the changes. Within even 6 weeks time period, you can see here in the cone diet, you can see these, most of the white patches. These are fat depositions, but we did not see some differences here in the lentil or the controls. So this is promising work, and we thought we would continue that. So actually pulses can help to reduce the obesity related things. So, and the next thing, so this is Nathan. So we have 2 graduate students who are looking at the what are the types of prebiotic carbohydrate, and then how its effect on the human health using the animal study. And then another graduate student who thought that, well, okay, then let's think about what happened, these carbohydrate, when you have it in the seed or the leaf, what kind of plant health mechanism. So this is one of the graduate students earlier did it. So we collected all these seeds from different countries, from ICAR. Dr. Kumar helped us. And then we found, based on the precipitation and the temperature, we see that prebiotic carbohydrates were changed. So we were targeted for sugar alcohols, RFOs, and some simple sugars. So we found that some of the countries has like high sugar alcohols and high RFOs with response to the moisture stress. So with that, Nathan decided to find out, okay, so is that the case? How about we all look at this pathway? So really, all these sugar alcohols, mannitol, sorbitol, they produce in the leaf and they export into the seed. So maybe that's why we are expecting this kind of correlation with the moisture and the temperature. So this hypothesis paper was published in the New Phytologist, new journal, like we published very recently, if you want to interested on that. So we think there is a hypothesis that in the leaves, these sugar alcohols were produced because of the temperature changes or the drought responses, and it will influence the seed nutrition quality. So that we decided to looking at the genome-wide association mapping. So we work with Dr. Kumar's program, and he gave us 2 mapping population, the global diversity panel, lentil diversity panel, as well as the heat tolerant population. So this data is still analyzing. So this is the heat tolerance mapping population. You can see some of the distributions of these lines, different compounds. So quite a bit of this is for Bascos and Castos, very nice distribution, and raffinose, tachios, because we are interested on these 2 and these 2. But we need to make sure glucose, fructose, sucrose are there, because these are the backbones to make all of these raffinose family oligosaccharides. So we went through and looked at all this, and this data will be published very soon. Still Nathan is analyzing it, but we see some genetic distribution and the genetic variation. So that means it is possible. And we also calculate the heritability estimates. The sugars that we are interested on the gut health, they have quite a bit of higher heritability. So that means we should be able to incorporate that into the breeding programs around the world. So right now, Nathan was working on the, I think at the moment, I can't say exactly, I think there's 7 to 8 QTLs were identified, and he is still on the process of the genetic, developing all the GWAS and identifying the gene pathway. Also, he has to do another gene validation study because we don't know yet everything. This is in progress, but it is promising. And I'm very impressed on that because this is a small thing that we start from the prebiotic carbohydrate about 2010. After 10 years later, now we know in the mapping population these traits exist and it is heritable, and then we go to the next step. So this is something that we've done in around 2010 to 2015 for iron and zinc and selenium. But we're moving forward now, the story to build up how low digestible carbohydrate would be helpful, not only for human nutrition, how it would be helpful to the plant. So Nathan's paper we're expecting to put out sometime in August. So I don't have much of the data to talk about that, but this is what we are doing. So a similar thing, we were working on the side-by-side for the, side-by-side for the iron, and most work done with the Dr. Shivakumar. And now we know modern cultivars about 50 to 75 parts per million, because concentration doesn't matter if it's not going to be bioavailable. So the wild relatives has a range. We have been screening so many number of lines, and then he has been incorporating this into his breeding program. And we look at the G by E and the heritability estimate we calculated. This is like a nutshell of what we have been doing simultaneously with the iron and zinc and selenium work and the carbohydrate. And also we discovered that phytic acid is the one that bind with the iron and reduce the bioavailability. We all know that. So most breeders wanted to reduce the phytic acid. But also in our research, we found that instead of the whole phytic acid pathway go up to IP6, if we can keep it there, IP3 form, and it would be more bioavailable. So one of the graduate students from my lab working on organic peas, and her project is to looking at this genomic regions in the diversity panel of the field pea, how this, instead of having phytate, if we can have IP3, that will be help to the plant to grow well as it is nutritious for the human. So we side by side work with the lentils and pea. And also we found out like the iron availability, bioavailability can be measured by Keiko cell cultures, which is done in Cornell University or Dr. Raymond Gland's program. But it is expensive, but we try to now screen with if something has high ascorbic acid and the target phenolics like two other phenols that we were interested on, camphorol, and there's a list of things. It doesn't come to my mind right now. I'm sorry. So we were targeting on these compounds and see the correlations between those and the ascorbic acid instead of we doing quite a bit of Keiko cell culture assays. So now we kind of have a model and we know, and when we have a mapping population or something, this is the range. And if you have this kind of target, so you can push forward. And I think this is incorporated into, and also we completed a human nutrition trial, and we know that 10% of the iron, if it is a 75 parts per million, 10% of the iron is bioavailable, which is already done. And ICAR, Dr. Kumar's program released more than 10 to 40 lines, and I found out yesterday it is not 10, it's more than 10, it's 40. And now we know all the biochemistry happening there. Now the next step is, can we develop a protocol that iron, zinc, and selenium can be measured in the field using the non-destructive tool. So that's right now we are working on it. So these are kind of things that I want to talk about, how phytic acid cooking influential, because when you cook, some of the phytic acid, like this is the pardina lentils, it's almost gone. So that's why I'm telling this, like cooking and utilization and how you process, how you store it. If you can incorporate these into the breeding program, that is very, very valuable. But I know it is difficult, but we have to somehow find a way to incorporate this kind of information into the gene pool to do more genetic work. So same thing for the selenium, which I have been working for a while. And now again, this is the screening population for the global mapping population, and we see quite a bit of ranges in the selenium, and then we can incorporate this into the breeding program. And we have done studies in Bangladesh. I think Canadian group actually done a study last year, human nutrition trial, Dr. Vandenberg's group. I think they fed lentils rich in selenium, and they are looking, he presented some data last year, which was promising lentil selenium reduced the acidosis in Bangladesh. So these are the things that we are doing. We try to because nutrition is a very broad thing. Like I said, in the food metrics, you see that how many components we have to work. And that's why we need help with all the genetics and anything we can get it. So, this is Dr. Kumar. Most of you know that I have been working with him, and it is really privileged to work with him and this variety that they released, and rich in iron, and we know it has high in ascorbic acid, and it has quite a bit of yield, as well as his work done in Ethiopia. And this is kind of an outcome if we can take it, and we know the genetics, and then we know the nutrition component, and we know what happened after the cooking, and then all these things going to be bioavailable. So that's a great success. And I want to talk about a little bit about this new project we started last year in November. It's the Feed the Future Innovation Lab. I want to go back. Previous. So this is the Feed the Future Innovation Lab for the crop improvement, established in Cornell University, with the partnership with the K-State and Clemson University, RTI, and the other partners from McRae University. And we established this lab for, this is the director, Dr. Stefan Krasovich. So the idea is improved varieties of key food crops are one of the surest avenue to reducing the risk of economic, environment, and climate shocks for millions of people. So our goal is to have these crops have the high food security and high nutrition quality, but this team has to develop tools to help the breeders around the world. So if you wanted to go to this website and follow it, it's all in there. And Dr. Krasovich is the program director, and from the Cornell University, he located in the Clemson University. So, which has which has 5 priorities. These are the areas of the inquiry leaders. So priority setting, and then trade discovery run by Dr. Jeff Morris, and the genomics by Dr. Buckler. And then our group is, we are working on phenomics, that how we can develop these tools, the chemical tools, into non-destructive way. Dr. Gore and Dr. Jesse Poland, they already have developed quite a bit of tools like PhenoApp, and then Dr. Gore has this handheld NIR to measure dry matter content and other components using the NIR. So my goal is to develop for carbohydrates, proteins, and some micronutrients, the chemical tools to doing into the handheld equipments to develop with Dr. Go and Dr. Poland. And then all these things going to Dr. Robbins' inquiry, that the breeder informatics. So if you are interested on, I think you should go on that website, because there's funding calls. A month ago there was a funding call, and you can see the updates on that. So I think it's important. So I wanted to give one of the small study that within 1 year, this is a case study that we wanted to show in the Feed the Future Innovation Lab that we could do it. So we decided, like my team, Dr. This is Dr. Darai, lentil breeder from Nepal. We thought, let's take Nepal as example. And can we develop a tool to look at the protein quality in the field? So working with Dr. Rai and Dr. Kumar, and this is Tava and the chemist, and also Dr. Krasavich helped us with the genome work. There's a quick study within less than a year. We want to develop this tool. So we already in the process, in the middle of the process right now in my lab, we are developing the tool, and we hope this will be available to ICAR AICRP-Pearl, all the breeding programs around the world by next year, spring. So why we picked the Nepal? Nepal is like 28.8 million people, and 57% of the population are working age, and 70% of them working on agriculture. And the worst earthquake hit in 2015 destroyed most of their agriculture lands and destroyed their infrastructure. So current nutritional issues are 40 to 60% anemia, and then the protein malnutrition, marasmus and kwashiorkor. Obesity is an emerging problem, and also food system. So the food system needs some help. So we as a group, we thought, how about we start there and figure it out? How can we do? I need to go back. So this is like for the collaboration between Clemson and the Nepal Agriculture Research Institute. We just established the MOU, and we try to start working with Dr. Darai, and this is his greenhouse. And the story is actually in this website, the web link, and you can go look at it. And we will keep updating our progress of the project. We just started, and actually we are measuring the developing the tool and validating it today. The engineer coming to my lab today to develop the tool. So objective of the goal is to develop a biofortified lentil cultivars with increased nutritional quality. The first thing is the protein. I think most of you know that pulses has low sulfur containing amino acids. So if we can develop a tool, the breeders can use that, pick the lines that high in sulfur containing amino acid in the field. That would be very valuable instead of doing a chemical phenotyping method, because chemical amino acid extraction takes 72-hour digestions and quite a bit of work. We are doing that in our lab using our LCs and the GC mass spec, and it is a process. It's a long work and hard work. But if we can convert that into the handheld way to look at the sulfur-containing amino acids, that is gold mine. So the clear objectives are the develop an undestructive tool to characterize the protein quality. In this case, we are going for methionine, cysteine, and phenylalanine. And also with the same time, we should be able to get the moisture and the starch and the protein from the same equipment. So today is a good day that because engineer coming to do the validation thing in the lab. So after this, I'm actually going to the lab for a little bit of time. And then we will validate this tool using our LC and GC mass spec technology. And then we will have a nice set of data that we can see the correlation. And then our goal is to test this in the field. And then we will send this tool. We will encourage people to hopefully this tool will not be that expensive. That's the other thing. Right now we have a commissioning tool which is about $20,000, which we use in the lab, but that's not the one we want breeders to have. We want to reduce it to quite a bit of less handheld equipment, so maybe then they can use that in their breeding programs. So this is the plots that Dr. Darai's field plots, and he planted those in 2019. And then we're using FTIR, Fourier transformation, infrared spectroscopy to measure this sulfur-containing amino acid. And don't forget, we're trying to do a workshop to train these breeders Dr. Tara is supposed to come and train, and then we try to, with the development of the Feed the Future Innovation Lab, we should be able to do these workshops around the world with the partnership with the PhenoApp group. I would like to give this technology to most people to use it. And so, like I said before, we're doing all these things. We have a really, really good group, genomic group, and the breeders, and my lab, and nothing possible without having a clinician. So this is Casey. He's one of my former students, and now he's a physician at the Mayo Clinic. So he also still interested on lentils. And our current work that we are working on is looking at how this lentils, especially we are focusing on lentils, and we are trying to see within the first 1,000 days how these change in the microbiome and how its effect on the weight gain and the obesity. So Casey working with a couple of interns and the students from my lab, and trying to figure it out, the planning the clinical feeding trials. That's what he passionate to do in his career with the Mayo Clinic. That is very prestigious. And also he works on vitamin B, and in Southeast Asia, that is lentil, one of the important crops there too. So we did quite a bit of work together. So if any of you interested, you can read all these papers. I'll share these slides with Dr. Varshney. So then the last thing with the time, this is our new platform, Going Organic. This is something that I'm very passionate about it. Going organic means, like, as I said before, pulses are very new to South Carolina. And we started a new project funded by NIFA USDA 2 years ago. This is a rotational crop breeding. So we plant pulses in November, December, around that time as a winter crop. And then we harvest it in May. And then sorghum go in around early June. And then that's harvest in end of September to, and then again, it's like a crop rotation study, breeding for the nutrition quality, but it is in the organic setting. We also do some breeding workshops for the nutritional breeding workshops, so you can in-house and learn from Dr. Varshney. I think I should do live focused because everybody can attend. It's a hands-on breeding workshops in Clemson, but if we do live, so most people can attend. So I want you all to go onto that website and find out like our workshops. And actually organic agriculture is quite interesting. I know breeders will say that it is difficult. It is difficult because management is very, very difficult. This is USDA organic certified land, and we cannot spray anything. It's all based on the management. But if you manage the field well, we are doing quite well. So there's still a lot to learn how we can incorporate nutrition component into that, because one of the ideas is the organic crops has low nutrition quality compared to the conventional grown one. So this is why we were looking into this. With that, so my final slide, Clemson, we developed the skills and insight that will contribute global food security. So this is what we are doing. My group, Dr. Krasovich group, we are working together and collaborating with Cornell, ICAR, and we all work together, and hopefully soon that we will work with ICRSAT. And this is our campus. It's beautiful campus. It's sunny, and it's right now it's like, it's very hot, and a couple of days it was cool, so it was good that we can harvest our peas. It's a beautiful campus if you get a chance to come and visit us. So with that, this is my team, my group, and they work very hard. And this is Dr. Shai, he's the breeder that do the selection with Dr. Kumar, and Krasovich and Dr. Boyles. They help us with a lot of genomic work, which is not my area. And we have quite a bit of funders, and all the work has done by collaboratively. And my lab is kind of in the heart of the middle and helping them with the nutrition and the phenotyping traits. And I hope to do this for a long time. So that's all I have. And thank you so much.
Thank you, Dr. Dil, for a very exhaustive and excellent lecture. Very good presentation. You have shown how the breeder should also start thinking from the nutritional perspective, and how the nutritional aspects also have to be integrated with the main core breeding program. And this is the call of the day. And we see there is a gradual shift in the breeders also incorporating breeding for nutritional traits. And moving ahead, you have also tried to show that your goal is to come out with a handheld instruments for good phenotyping at a low cost and save the time and resources and energy and labor. That's a very good goal, and we hope that you'll come out with that in the near future, because we are also selfish, because we also want to use those things in our breeding programs. As a pearl millet breeder, yes, we have been breeding for high iron and zinc. And as Dr. Rajeev has already mentioned in the beginning, that we have come out with minimum level of iron and zinc in our cultivar release policy. This is the first of its kind where we have tried to integrate nutritional quality into our mainstream breeding program. So we hope that such things happen in near future in other crops also. In lentil also, I see that we have come out with very exhaustive information which can be tapped and used by the lentil breeders across the world. I think many students and many researchers have many doubts. I have seen them flowing in, and Dr. Rajeev, the floor is yours.
Good. Thank you very much, Tara Madam, and thank you very much, Dil, for great presentation, very comprehensive. And there is one comment from Dr. Okole, and I would like to add a few things on that one, and this basically summarize overall your work, and we are so impressed. He says, and I will add something. We like the way you have integrated nutrition and gender, and I will add the genomics and breeding in your work. Having a product profile, having those key traits, including in the crop improvement agenda, would improve crop improvement and make the end product more acceptable and impactful. Having a team of nutritionists, social scientists, I will already add the breeders and geneticists and bioinformaticians working together with your team will complete the chain. We need desirable genes to improve adaptation, high yields, which are also nutritious. So you are doing remarkable work. So congratulations then. So I think
thank you.
Very nicely. Yeah. I added something from my side as well. You are there. Yes. Good. So thanks a lot. Now we know that there are limited time, but still we would like to have some questions from, we'll take some questions from Zoom, some from YouTube, and I can tell that around 1,500 to 2,000 people were watching your presentation then. And one person says, I'm not a breeder, but a food and nutrition scientist. We recently found that some wild legumes contain higher minerals like iron than some domesticated one. So do you think that domestication of the wild species could be a good line of research, or it is better to use them in breeding domesticated ones? Your advice?
I did that for selenium, because when we look at the selenium germplasm, most high lines were in the wild types. And actually they take up quite a bit of selenium compared to the adapted lines. So it is up to the breeders. I recommend to go all, and we need to have as much as the germplasm variety. Like if selenium paper, you can go, I think it's in the plant journal. There's like so many number of genotypes that those are wild and adapted. They all came from Dr. Kumar's program, Shiv Kumar's program. And we see wild types has the most, and it's sometimes because they were there in the Dr. Varshney can help me up with that. This is the evolution process, right? Yep. And so based on the soil or where the harsh times that they were growing in, so they have adapted to that. So if we can get their valuable genes into the adapted lines instead of taking non-valuable ones, I think that is so important. That is a question like someone like Dr. Varshney can help us with to get those genes into the pool, which is that we are doing for selenium as well as iron and zinc. I can't comment on the carbohydrates yet, but I have a feeling that amino acids going to be quite changed in the wild lentil collection. And which we will have data. I am hoping to give all this data to Green and the USA database, so everybody can access and go pick the accessions. Because I want all the breeders to use this.
And Dil, as I mentioned earlier, and just for the audience, so we have the colleagues from about 67 countries, and many countries they're from Africa. So some colleagues from Africa, they have made this point, and one of this very nice point, they say that we need that kind of breeding for the nutrition in Africa. So we are really looking forward to see its work, and we hope that your work is going to make the impact on our program. So this is really good comment from them. And one quick question that they say, one person asked that, do you know if there is an interaction between polyphenolic compounds and carbohydrate absorption? In other words, can polyphenols reduce the glycemic index?
Yes, yes. There's quite a bit of literature in it. Carbohydrate and polyphenol interactions. We haven't studied that, but it is something in our plate to study, because studying polyphenols is quite a challenge, I would say. Right now, we only do target measurements like camphorols and some of the ferrolic acid, the target ones for the iron bioavailability. But this is, see, I'm telling you, like, this is never ending process. So if you focus on carbohydrate, all the machines are working on carbohydrate. So we have no time to do polyphenols. But with the approach of metabolomic approach, I think we might be able to find out the connection. But definitely there is a connection, and bioavailability is governed by the secondary metabolites. So we have to look into that. That's a very good point and a good question.
Good.
I'm glad that they understood.
So other question is that RFOs are generally cause flatulence, hence framed as anti-nutritional factors. I'm curious to know about the role of RFOs in human gut health and how much is the right concentration to have in human body.
That is a very, very good question. So that's what I said in the earlier of my presentation. Decades ago, breeders actually trying to reduce the RFO, especially for chickpeas, lentils, peas, because of the flatulence and the gas. It's kind of a problem. But now we are seeing more literature. This RFO is one of the good substrate for this bifidobacteria, which is proven by our rat study. But I can't tell you exactly how much, because working with Casey, we are trying to understand this question, because if you grow up in a country eating lentils, or like after your 6 months or 9 months, your first diet is pulses, your gut microbiome actually adapted to this kind of food, right? But if you don't, like the Caucasians, like here we're in South Carolina, so if you don't grow up eating with that, you have a difficulty on digesting and the flatulence. So you have to start this slowly and work it out. So we still don't know exact how much, but you cannot knock it down, because it helps to keep the plant health as well. So I should be able to give an exact answer probably within another 3 months. We can tell you exactly how much for the lentils, but then someone else had to work on the other crops and see how much in it.
Okay. When you mention other crop, then there is a question about rice. They say that what is the role of prebiotic carbohydrate in case of rice crop? Just want to know. I'm doing my research on nutraceutical and micronutrients properties in rice. So I would love to take some opinions and advice from you, madam. And yes, it's really high time to collaborate, to have the collaboration between quality breeders with the nutritional scientist.
Well, I would love to, we can personally talk about it. Like the rice has quite a bit of info, like the sticky rice mean how the way you cook, you have quite a bit of resistant starch coming out. So if you have resistant starch, that means it's good on your gut health. So whoever that person, I would happy to help. And actually, Tava did one of the study with adding some coconut oil when you cook the rice, or like adding coconut milk. It changed the range of concentration of the RS, which help on the gut microbiome. So rice is also like one of our main crops, right?
Yeah.
And I am not working with it, but I'm very happy to help.
When you say about non-working, and uh, although you have been a great friend, but I always have this complaint to you that you don't work on chickpea and pigeon pea on our crop.
I do work on chickpea with Dr. Vandermark. I just only present this for lentils and pigeon pea.
Yeah.
With the Future Innovation Lab, and I really want to work with Dr. Varshney as well. Like, I don't know, I have been working with Dr. Kumar for like
Yeah.
decades as a grad student.
Although Dr. Kumar is also a good friend, but no, nevertheless. So, well, anyway, so now there was a question on pigeon pea. That's the reason I also raised this point.
Right.
And this pigeon pea researcher says that we worked on agronomic fortification to enrich zinc in pigeon pea with application of relatively more dose than recommended dose. Got good results in increasing zinc in grains, which is encouraging. However, zinc content in the grain was low when the crop was grown on the zinc deficient soil.
Right.
May I know your opinion, please?
Oh, that's a great thing. Everybody knows about Dr. Chuck Mack in Turkey, that their wheat was they found that the stunting and the growth, and when they add zinc, and this is very well known. I'm very impressed, and I think it's so we can do the same thing for selenium too, but is it economical? That's the problem. So breeders might not want to do this all the time. So if you can, and they have a high heritability, so I think we should be able to breed it, right, Dr. Varshney?
Sure, I think so. And anyway, so I think there are many questions we cannot take now, but nevertheless, we are grateful to you again for very great presentation and to create or to excite everybody here, especially in terms of this nutrition and then combining the different areas. We are very much impressed, and we look forward, especially I'm looking forward to working with you, Dil. I hope that
Thank you.
sometimes you will show my picture also in your slide. So I'm looking
Of
forward.
course. Of course.
Okay.
Thank you so much.
Yeah, thanks a lot for joining us. And again, thanks to get up so early in morning and to join us. Thank you very much.